this case. Okay, it's EGFP, meaning green fluorescent protein. Okay. So now we ask what happens. So the first thing we did was turned on that fusion gene in every cell of the body. And that's HPRT is expressed as first cell division and continues to be expressed. So every cell in your body has that fusion gene. What happens? And what happens is shown here. You get a green blob. Okay, that's good because we're turning that fusion gene on and then those cells become green. But this is a little embryo, and what we can see is it's a completely disorganized tissue. And then it gets very quickly absorbed and doesn't give rise to a cancer. And that gives us some information. That tells us that this gene that's turning on and off genes at random now is very cytotoxic. It's does very bad things to that embryo. And so it starts to develop new tissues. It's completely random and it dies. And it's reabsorbed in a couple of days. Okay? Doesn't give rise to cancer. So now the next thing we decided is, well, let's try muscle. Okay, why muscle? Because the other thing that's present in every joint in your body are also muscles. You have to be able to move those joints. Okay? And so we looked at to make muscle requires a number of steps. You start out with progenitor cells, you make myoblasts, you make myotubes, and then they make the fully differentiated muscle fiber that allows you to contract and expand joints and so on. Okay? And so that's what we did. So I showed you the first one, HPRT, those embryos died. So the next time we went is all the way at the bottom, fully differentiated muscle. And MIF6 is a gene that's expressed only when you have fully differentiated muscle. Okay, for example, in this muscle right here. Okay, and SM2 mouse is the mouse that has that construct that I showed you, and Cree is being controlled under MIF6. So in fully differentiated muscle, that protein is made, and we can ask what happens. And this was interesting. It didn't give rise to tumors, but it gave rise to a myopathy. And a myopathy is simply a disease of muscle. And there are many different myopathies. And here I'm showing you muscle. This is normal muscle. So this is looking longitudinally down muscle. This is cross-section of a muscle. And these are the muscles in the presence of this fusion gene. And what you can see, for example, instead of having nice straight fibers, they're called wavy fibers. These little purple are the nuclei, and they're all lined up in a row. That's not common in normal muscle. Okay, and what that tells us is that the muscle cells are dying and then are being replaced. If you, for example, exercise too much, then you have a system that will then repair that muscle and make new muscle cells, and then your muscles are still okay. Okay, and these are called satellite cells. And typically, when you have a repair of muscle by satellite cells, then all the nuclei are lining up. So what that tells us is that the muscle cells are dying in the presence of that fusion gene and then are being replaced by satellite system. Okay? However, these mice didn't develop tumors, and they eventually died of the myopathy. Now, why this is interesting is that I told you that there are 10 to the 14th translocations in your body. Now, in cancers, we know that a translocation happens because in that cancer, that cell is expanding, so you have a whole mass of cells that have a common cell of origin. Okay? And so if you see something wrong in it, then it's easy to see. Okay? In other tissues, if you don't have that expansion, you will never see essentially what's happening. What we're saying is that a translocation, specifically this translocation, can give rise to myopathies. And in fact, recently, a myopathy has been identified with a specific chromosomal translocation. And so what I'm saying is we should be keeping in mind that now translocation may start creeping up in other diseases that we never suspected might be as a cause of a translocation. Even, for example, neuropsychiatric diseases could be as a consequence of a chromosomal translocation. And here's an example where it gives rise to a myopathy. But it didn't give rise to cancer, so then we have to go back. And so next we look at muscle progenitor cells. This is another gene that's expressed in progenitor cells, PAX3, PAX7. Wherever those genes are function, Cre is turned on, and the fusion shoot is made, and what happens is the embryo dies and no cancer. So finally, we went to myoblasts. Myoblasts are a step that are just before the fusion of all those cells to make the myofiber. And we could have chosen one of two genes that are important for myoblasts. One's called MYF5 Cre, and the other is called MyoD Cre. Both are important for making myoblasts. So we made MYF5 Cre, me meaning Malay Haldar, and then we crossed into our mouse and then asked what happens, and what we got are tumors, and we got synovial tumors. Here we show that they arise near joints, as expected, and they always arise near joints. And then we have now many, many criteria to say, are we really making a tumor that's very similar to human synovial sarcoma? And for example, it metastasizes, and we can follow that because those cells turn green. And so we can look, for example, lymph nodes, and here we see a green tumor starting to arise, and that is metastasized. It's a small tumor that's coming from the big tumor. And here it is in the cerebellum, in the brain, and again, there's a metastasis starting there. Okay? And the pattern of metastasis of this tumor in mice and humans is identical. Okay, here I told you about histological types. And what we found is that they're both what we call simple spindle cells and also more complex biphasic. And we see the same thing in the tumor, in the mouse. Further, if it's SSX1, it's normally mostly biphasic, and if it's SSX2, it's mostly monophasic. And we see in the mouse a ratio of 10 to 1 being monophasic versus biphasic. There are many, many genes that are specifically expressed in this tumor, and here's simply a list. There are over 25. I'll just show you a couple. These are genes that are found in biphasics. These are skin, and here we see, for example, CAM5.2 is expressed in the gene, cytokeratin is expressed in this tumor, also in the humans. Myogenin is interesting because myogenin is a gene that's usually expressed in fully differentiated in muscle, and it's not on. Okay, so this one, it doesn't show this expression. You don't see this brown color. And the reason that's interesting is that all the genes that would tell you to make muscle are now turned off by this fusion gene. And so that's why it was never recognized as being a muscle tumor. Okay, BCL2 is an important gene. Over 99.5% of the tumors have BCL2 positive, and also this tumor has it, and so on. And in fact, now we can look at thousands of genes and in fact, people have looked at many, many tumors in humans, and each gene, each tumor has what we call a specific fingerprint, which is thousands of genes specific to that tumor, and tells you, and if you look at many, many tumors, one type will only have this fingerprint and another will have a completely different fingerprint. And by a fingerprint analysis, where we're looking at thousands of genes, again, the fingerprint's the same as human synovial sarcoma. So we know that this model is now really simply very similar to human synovial sarcoma. Now, one of the things that was surprising is shown here. This is the tumor in surrounding muscle. This light green is what we call autofluorescent. All muscle tissue, if you shine UV light on it, it autofluoresces and gives you a